Thank you everyone for joining us tonight. Our topic is about drug development, the potential for future therapies for KAT6 disorders by Typhaine Lejeune. A short biography about Typhaine. She is a doctor of veterinary medicine and veterinary pathologist with a specialization in anatomic pathology. Typhaine has been working in Canada in the pharmaceutical industry preclinical field for the last 13 years doing drug safety assessment. And she has a 6-year-old boy with a KAT6 disorder. Thank you, Typhaine, and welcome everyone.
Thank you, Emile. I'm very happy to be sharing this with you tonight. So I thought I would actually start with a small introduction about drug development, just for you guys to understand what I'm basically doing here tonight and why I am presenting this. So drug development is long process, usually 15 to 20 years for a drug to be approved. It starts with a very high number of compounds that are being tested to see if they are efficient in certain areas. And then, as you can see, it slowly goes down to very few number of them in the end. And the field I work in is actually this one, preclinical development. So you can see that already there are a lot of candidates that have gone through to the trash directly. Still, there are quite some of them left, and those are being evaluated notably for their toxicity, their efficiency. Sorry for the cat. And very few of them really only go until clinical development, which is the phase where compounds are tested on humans. So obviously you want to be careful before giving anything to anybody. So those are the steps. And as I was saying, I work in this preclinical environment where all these things you don't need to read are evaluated. As you can see, safety and efficacy here, they are also evaluated during the clinical development. Part of the clinical development is occurring while the preclinical is still ongoing as well. But safety assessment does occur mainly during preclinical development. And it is mainly to evaluate the safety and the toxicity of drug candidates to determine what the first doses will be for people in clinical trials. And as a pathologist, what I see is I evaluate this toxicity at the microscopic level. So this is what I do every day. And as such, as you can see, a lot of drugs are going to the trash, which means I see a lot of different compounds that are not yet approved for the market. So I get to see the trends, let's say, for future, the new technologies that are being developed, the new types of drugs that are being developed. Mm-hmm. And I have seen this for the past 13 years. And over these years, I have seen one specific class of drugs that is becoming very big now, and the first ones are being approved currently. And this is what I wanted to talk to you about. So back to basis first I think, to the genes, their structure, because I don't know what you guys know, so I'd rather go back from the beginning. And if anything is not clear, please just stop me and ask me a question. So within the cell is the nucleus, and this is where our genes are. They are in the chromosomes. We as human beings have 46 of them. And chromosomes basically usually have this same sort of like X form, and they are composed of tightly packed DNA. The DNA is enrolled in there and very, very, very compacted. And then whenever there is need for a specific protein to be synthesized, the DNA needs to be unrolled. And that's where you can see the full structure here, this double helix. And these letters that you probably all know by now, the C, the G, the A, the T, those are the bases that compose the DNA. So here is the double helix. Very quickly, there is this blue here that we call the backbone, because this is where the bases are attached. And then those bases, they always associated in a paired manner. A always associates to a T, G always associates to a C. It's always like that. And that's how the genes are made. Then how are proteins synthesized? Whenever there is need for a protein to be synthesized, the DNA, the gene of interest, is being transcripted. And that means a single strand instead of this double strand that you saw for the DNA. A single strand is being produced, and that is the RNA. And this is the single strand that comes out of the nucleus here, and then it will be translated into a protein. For this, each small sequence of 3 bases that we call a codon will equal to an amino acid. And so each amino acid is added to one another and ultimately will form the protein. So the RNA here is really what is of interest to us today. So some words about KAT6A and KAT6B disorders. Almost all our genes come in 2 copies, one from each parent. So if you take this chromosome, which we call wild type, meaning what is most common within the whole human population, if you cut it in half, then the left half came from, let's say, the mother, and then the right half came from the father. And that's how you get 2 copies of this gene, which is called A here in that example. Then in KAT6B disorders, those disorders are called autosomal, which means simply means that the genes are not on the sex chromosome, the X or the Y. And then they are called dominant because only 1 copy of the gene bears the mutation. So this is the example here of a mutant variant. 1 copy has the mutation, the other copy is the normal one. And so that means that there is always, in all our kids, whatever their variant is, there is always a normal copy of the gene that produces a normal protein. And then the issues arise because of this other copy that is the mutant variant. And either it ends up in the synthesis of an abnormal KAT6 protein, or even in some cases also in an absence of protein. It just depends on the position of the mutation within the gene. So oligonucleotides are this new class of drugs I was talking to you about. They have actually been developed since the 90s. I have always seen them under my microscope since I've been working for the past 13 years, except I have seen more and more of them over the past, let's say, 5-6 years. The first one was actually approved in 1999, and several were approved over the past 4 years. So those are all the ones that are currently on the market. There are 11 of them, as you can see. I will talk to you a bit more about this one later. But as you can see, they all relate to genetic diseases. And those are quite frequent. There is just one exception here, the spinal muscular atrophy, that is a rare disease. And that's where the Milazan came. And I will come back to that story a bit later. And so those are the ones on the market currently. And there are other pharmaceutical companies that are actually currently fighting for new ones. So really other ones are coming in, and also other ones for rare diseases, which is very interesting for us. So what are they? Oligonucleotides are actually very similar to RNA, meaning that they are a very small sequence of bases, usually between 20 and 50 of them. And the only difference, because you cannot actually, it would be too difficult to make RNA and have it actually survive within the cells. So the only difference is the backbone. The backbone is just chemically different from RNA or DNA, but then the bases are there. And that's what matters, because they are, each of them is designed to specifically match an RNA sequence. So for example, for us, what is interesting is that it can match a specific variant of a gene, of part of a gene. And so since they match that sequence, it means that with the base pairing I was talking to you about, the A with the T, the G with the C, the oligonucleotide, which is designated ASO here, will bind to the messenger RNA. And that's really a very interesting thing, because what can happen from this, depending on the sequence that is the target of this oligonucleotide, its binding to RNA will induce two things. Either it will induce the destruction of the messenger RNA and result, no protein synthesis would be a great thing, but in real life, it doesn't result in really no protein synthesis, but actually less of it, which is already something very important. And over the past few years, there are new generations of these oligonucleotides that currently, if I remember well, we are the 4th generation of them. And a new function has actually arisen, and that is that they can modify the RNA and therefore modify the protein. That means, for example, a protein that was inactive could become active, or a protein that was toxic could become non-toxic. So that's what those are the kind of things that oligonucleotides are capable of doing. So now for something a bit more concrete, I will just talk to you about two stories. So first, the medicine story that I was talking to you about in the beginning. This one is the very first patient-customized oligonucleotide, and it happened in 2018, actually, not 19. Mila Mila was diagnosed, was a young girl, a young 6-year-old girl that was diagnosed with a unique variant that was never identified before of Batten's disease, which is a disease. This disease is a neurodegenerative disease with symptoms starting around the age of 3, and the life expectancy of these children is actually just over 10. So this girl came to the hospital, and she was 6. She was losing her vision. She was becoming more and more clumsy. She was losing her work. The Children's Hospital in Boston diagnosed her and identified her mutation and decided to design a unique oligonucleotide that was customized for her specific mutation, meaning that this drug could only be given to one person, not any other person in the world, because she was the only one to have this variant. And so the clinical trial, what is quite amazing with this story is that the clinical trial occurred within a year following identification of the mutation, which is highly fast for a drug to be developed. And so this is here, I took the timeline from the article just to show you how it happened. She was diagnosed in November 2016, and then the Dr. Yu identified her mutation in March of 2017. Then he identified a proper oligonucleotide in September, and they started doing the first preclinical toxicologic study in rats in December, and she was administered her first dose at the end of January in 2018. So this is really very fast. And I can tell you, because I am pretty proud that my company took a role in this, because it was one of my colleagues that actually stayed on throughout December, including December 25, to look at the slides, the microscopic slides of this study, because we knew that this girl was waiting for her first dose of this drug, and it was really urgent for her. So this is a very specific case, of course. They were able to go that fast first because there was a real urgency for the patient, because she was deteriorating. Also because this doctor based his oligonucleotide on nusinersen, this drug that was already on the market and that was being administered to infants safely. So he used the same backbone, the same chemistry, and he changed the sequence of base. That's what he did. So obviously, he already had an idea of the safety of this drug. So that's why it could go so fast. So it's not desirable for KAT6 kids because KAT6 disorders are not degenerative disorders. So the urgency is different because this approach still bears some risk, of course, going so fast. But I just think it remains a very good example of the possibilities that are out there with these compounds. Then the second story I want to talk to you about is Angelman syndrome. This one is a bit more similar to KAT6. It's a rare neurogenetic disorder, so it's not degenerative. It's caused by a mutation in the gene UBE3A, whatever its name is, but the mutation is there. It occurs spontaneously in most of the kids. And how this gene works is that in a normal, every individual, the copy of the gene coming from the father is actually silenced. That's just how it functions. So the copy coming from the father is just not functioning in everybody. And then the maternal copy, if it is either missing or mutated, then the Angelman syndrome will occur. So the signs are developmental delay, or impairment, seizures, speech difficulties, feeding difficulties. So I'm sure it reminds everybody here of something. And so the oligonucleotide was actually the target of the oligonucleotide that was created for this one is forcing the expression of the paternal copy of the gene to restore at least part of the function. And so this one is actually went through a more conventional, I would say, path, for development. They did non-clinical studies before. They did studies on cultures of neurons, and they could see that the paternal gene was actually reactivated. They also got mice with Angelman syndrome, to which they administered their oligonucleotide and saw improvement in some of the symptoms. They also got results suggesting that administration of this drug was not only for kids, it could also rescue part of the phenotype in adults as well. So that's very interesting, obviously. And so two mothers with kids with Angelman syndrome decided to create a company to develop the compound. They were able to do so thanks to a foundation that exists for Angelman syndromes, a biopharmaceutical company that is specialized in the treatment of rare diseases. So then their compound, GTX-102, was granted orphan drug designation and rare pediatric disease designation from the US Food and Drug Administration. I just put this in here just also to emphasize the fact that these drugs are new, and especially for rare diseases, the regulatory authorities that approve the drugs, they had to adapt because these drugs cannot follow exactly the same path as any regular drugs, because a lot of them are directed to kids first. Clinical trials, usually the way they happen, there is the first phase where you give a drug to just healthy people. And this is not useful for such drugs. And then usually you have clinical trials with a placebo group that won't receive the drug and a treated group that will receive the drug. But obviously this brings, when you have very few children and they all have rare diseases that are quite disabling, it obviously is not ethical to have a placebo group. You want to give the drug to all the kids. So all this to say that these agencies now, these regulatory authorities have adapted to that over the past 10 years. And so there are specifics for these drugs. And so the clinical, back to Angelman syndrome, the clinical trial actually started last year in March with 5 patients. Obviously, there are always very few patients in these clinical trials. And the safety assessment, the preclinical studies are actually still ongoing. And so far, they actually announced the first results, and they see really substantial improvements in all patients. So this is really very good news for these kids. So now what for our kids, the future for KAT6 disorders? Millicent and Angelman syndrome, they are definitely not unique. The stories are still rare, but there are still similar ongoing studies for other rare diseases. And I can tell you because I see them on a regular basis. And they just illustrate, I think, all the possibilities and the hopes that oligonucleotides bring, I think, for rare diseases in particular. And what is good is that, as I was saying, they have been developed since the 90s. So the safety profile of this class of drug is now well known, which can facilitate the development of a specific molecule. Obviously, fundraising remains the key because obviously to develop something, you need money. And then specifically for KAT6, that means that oligonucleotides could be customized really specifically for a designated variant. So obviously the challenge with KAT6 is that there are many, many, many different variants, but still, I'm sure one day oligonucleotides will be probably, they're already quite easy to make actually, but they will be so easy to make that they will be able to customize them for every single variant. I'm pretty sure of that. And so that means that KAT6 kids with an abnormal KAT6 protein that is synthesized could potentially be improved if this protein was shut down, less sensitized, and then the normal protein would remain, and this would potentially result in a milder phenotype. In some other variants, I think it is all a matter of variant type, of course, but in some other variants, maybe some of these proteins could actually be modified in order to restore a fully functioning protein. I think everything is imaginable, honestly. And so most of the current research focuses on the mechanisms at the molecular level of KAT6 enzyme function and the role of KAT6 enzymes in cancer, notably, which is great. We need to have that kind of information, but I think that also to benefit the families in a more direct way, probably part of this research could also focus on such molecules and see what could be done with them. So this is my own KAT6B little boy, just to say thank you, and I hope that was interesting to you all. So if you have questions, please do so.
Thank you, Typhaine.